to the last Soil, Water, and Climate Department seminar of the semester. I'm Kelly Wells. I'm a graduate or research associate in the department and a co-organizer of the seminar series along with Vasudha Sharma and Kyle Sherbine. And so I want to thank them for their help in putting together the series this semester, and just want to say if anybody has any feedback on this semester's seminar series, or if they have recommended speakers for next semester or further into the future, please reach out to one of us. We're also always happy to hear your thoughts. So, and also want to give a big thanks to Amy Lesby for her help in communicating the series and also wrangling all the technical aspects of things. If you missed any of the previous talks this semester, all the recordings are online on the department website, and I think Amy sent out a link to that in her email today, so please go and check those out. But today I have the pleasure of introducing our speaker, who is Tammy Walsky. Tammy is a MS candidate in the Water Resources program in Dr. Satoshi Ishii's lab. As an environmental microbiologist, she aspires to understand the roles and functions of microorganisms and how they intersect and impact human and environmental health and quality. She earned her BS in biology and in environmental science and technology from the University of Maryland in 2015. And she's originally from Baltimore, Maryland, and did not know anything about Minnesota before she moved here, but she says she remains very pleased with her decision. So thanks so much, Tammy, for being here, and I'll leave it to you to get started.
Thank you for the generous introduction, Kelly. Hello, everyone, and thank you for coming. I'll present and discuss results from one of my years of research for 2019. Temporal dynamics of pathogens and antibiotic resistance genes and raw and treated stormwater. So why reuse stormwater? Reusing stormwater can reduce demands on groundwater aquifers and improve surface water quality, especially as precipitation events become heavier and flooding becomes more common due to climate change. Stormwater reuse can play a role in reducing flooding in areas where there is a lot of impervious surface. However, public perception of health risks associated with microbiological contaminants remains a key barrier to the expansion of water reuse, as well as a lack of knowledge around the general microbial contaminants of stormwater, especially here in Minnesota. Much of the research surrounding this has been conducted in drier, hotter places where water reuse is a bit more of a urgent need, and that ultimately has limits as to what is applicable to here, which is where it's kind of the opposite, you know, generally colder and wetter. So to measure microbial contaminants and quality in water, we generally use fecal indicator bacteria. E. coli is the primarily one used, though enterococci and streptococci have also been used. However, there's only limited correlation between fecal indicator bacteria and pathogens. There are many pathogens that can be present in water that are not fecally associated. Viruses, especially norovirus, are also waterborne. Protozoan or single-celled eukaryotic organisms, it's like Cryptosporidium and Giardia, also infect people and are naturally occurring in waters. And then antibiotic resistance genes are generally considered more of an emerging contaminant, and increasing antibiotic resistance across the world is an alarming health concern. While antibiotic resistance genes naturally occur in the environment, human factors and human use have definitely impacted their occurrence and proliferation. In a previous study that the Ishii lab did, they sampled at 25 water reuse sites as well as wastewater treatment plants across the state. But they only sampled at the sites once, not taking into account seasonal fluxes and the variety of factor that might affect the results. Plus, out of the thousands of liters of water used, due to our sampling methods, only about 26 liters of water gets filtered through our filter, which then goes back and becomes the DNA that we use in our research. 10 different pathogens total are found at these 25 different sites. In general, E. coli levels didn't correlate with the chemical data or pathogen data at all. So in order to draw conclusions, we needed more frequent sampling. Project objectives include monitoring levels of pathogens and antibiotic resistant genes in stormwater samples, evaluating effectivity of water reclamation systems, and analyzing correlations between pathogens and antibiotic resistance gene levels, and environmental and physiochemical parameters. So environmental parameters like water temperature, precipitation, and physiochemical parameters such as pH or turbidity. What environmental factors influence pathogen and antibiotic resistance gene levels? Can we predict the occurrences of pathogens and antibiotic resistance genes in stormwater based on environmental and physiochemical variables? This study will significantly advance our understandings in how pathogens and antibiotic resistance genes occur in Minnesota stormwater and how well these contaminants can be removed during the stormwater reclamation processes. So will help, this will help Minnesota stormwater practitioner better manage stormwater, as well as give the Minnesota Department of Health a large data set to work with in terms of determining general risk and general risk of using stormwater in various situations. So a bit about my sampling, collection, and processing. I sampled from 5 different sites that reuse stormwater across the Twin Cities metro area from June to October 2019. Of these sites, only 2 had microbial treatment, and I also sampled from a site that uses tap water, or like the chlorinated municipal city water for irrigation as well. Out of the 2 stormwater reuse sites that had microbial treatment, one site had chlorination and the other site had an extensive treatment system with UV light and multiple filters. All sites had some sort of sediment filter to filter out debris and particles. Each system is set up differently, and we sampled from different points within the stormwater reuse system, generally taking a sample from the input water, a sample during or right after treatment before it reaches its final destination, and a sample at the output, generally an irrigation head or hose outlet. We filter the water through a dialysis filter as seen on the left-hand side of the slide. Once we were finished with that, the filters were taken back to the lab, back flushed for recovery of DNA and microbial cells. The back flush is then flocculated, centrifuged, and pelleted. And from there, and then from the pellets, the DNA was extracted. Aside from molecular data, we also conducted the coliform test in order to collect coliform forming units and E. coli data. The coliform test is a very easy, commercially available assay used to monitor total coliform and E. coli levels and can be used daily. The whole process itself takes 24 hours. It provides the most probable number, or MPN, of units per 100 milliliter of water. So the left-hand side has coliform forming units. On the very left of the picture is a site, or is a sample that had no coliform units. And then on the right side, you see the little packet with the yellow. Each of the little squares that's yellow stands for a different amount, and you add up the total squares and then get the most probable number. On the right-hand side, you have E. coli, which is done when you place the whole little slide under a fluorescent light, and the little squares that have E. coli fluoresce. Including this in our study allowed for a comparison between these results and then the more detailed molecular data. In the microfluidic quantitative PCR system that we used to obtain the molecular data, multiple quantitative PCR assay reagents are mixed with sample DNA in nanoliter volume chambers that are present in high density on a chip, providing high throughput qPCR reactions. This system reduces sample time analysis and reagent requirements when compared to conventional qPCR, which, done on a standard 96 well plate, can only take about 88 samples plus your standards, and then only quantifies one gene at a time per plate. For this study, I quantified 48 antibiotic resistance genes, 23 bacterial genes, and 13 viral genes. So before we jump into my results, I'm gonna clarify between two similar phrases that I used for interpreting my data. When I say number of detections, I mean the total number of genes detected in the sample. It's either over the course of the sampling season or for a particular sample. So an example of over the course of the sampling season would be site A output. So I'm giving both the site and then the particular place where I sampled from had 10 detections over the course of the season. Per sample, I would say sample 1 had 5 detections total. As in that particular day, time, and place, 5 genes were detected. When I say quantity of detection, I mean the gene copy number per liter of water of the specific gene on a specific day. So, for example, sample 2 had 3 gene copy numbers per liter of water for TET-A, which is a gene that encodes for tetracycline resistance, on the 1st of June, 2019. As for an overview, antibiotic resistance genes were more frequently detected across all samples than pathogenic bacteria and viruses. And no viruses were detected in any of my samples. Only 3 bacterium were detected: human adenovirus, general E. coli, and Shigella flexneri. We detected a lot of general E. coli, 14 times. It's a very common bacterium, and that's not surprising at all. Human adenovirus was detected 5 times in 5 different sites, including the site that had chlorine treatment and the tap water control, and the site that had extensive treatment. Shigella flexneri was detected 2 times in the same system. Shigella flexneri is an enteric pathogen that causes shigellosis and is usually spread via contact with feces or by contaminated food and water. Although there are pathogens that were not included in our testing suite, E. coli and total coliform detections did occur where no pathogens were detected. In particular, the 2 samples where Shigella was detected had 0 results for most probable number for both E. coli, and then less than 1 for most probable number for total coliform from the collar assay. Over the next couple of slides, I'm going to elaborate on what I did to look at the data set as a whole. In this particular slide, you see a box plot of the quantified gene copy number of the 16S bacterial ribosomal subunit for each DNA triplicate. I extracted each sample in triplicate, then ran 16S for each of the triplicate, and then averaged them to get 1 16S gene copy number per liter of water per sample. Using this data, I made a box plot sorting by samples to compare the total bacterial presence in each system. You can see a difference between the tap water and the St. Paul site, which is the site that had extensive treatment, and then the rest of the sites. Though this difference is not astronomical. Looking at the mean values, the other sites are about 0.5 log gene copy numbers per liter of water, higher than the St. Paul site. Oh, actually, my bad. This was the gene copy number data. So this was the 16S. You can see the St. Paul and the tap water are a little bit lower. And then this is the gene copy number, and it looks very similar to the 16S data. This includes the detected gene, antibiotic resistance genes, and the detected bacterial genes. Once again, there's a difference between the St. Paul and tap sites and the other sites. In this case, the mean of the St. Paul site and the tap water are 0.5 to 1 log lower than the other sites. The St. Paul site has some notable outliers, but the gene copy numbers per liter of water are overall pretty compact. This tells me that overall less bacterial presence in the treated sites, and then overall quantity of the detections is less in these sites as well. So then I took a look at the number of detections across the whole seasons for the St. Paul site. Because the site is treated so extensively, we pulled samples specifically generally after each round of treatment. And I wanted to see if there was a decrease between the first sample, which was after the first round of treatment, and then the last sample, which was at the output. Looking at the data, I do not see a general decrease as you go from the first sample down to the fourth sample. The differences are generally by date. And then each of the samples in each date are, for the most part, around the same number of detections. It was not different enough to make me want to run any statistics on it. So for this particular site where it has that first round of treatment, it's telling us that the subsequent treatments are not doing such a more drastic job in making the water cleaner, so to speak. For the next part, I made a correlation plot of all the environmental and physiochemical factors, along with different components of the microbial data, including molecular data like 16S values and gene copy number, as well as call alert data. Overall, there were no strong correlations between physiochemical and environmental factors and microbial factors. The one strong correlation was between 24-hour and 48-hour precipitation. Other than that, the highest correlation value was 0.65 between the log gene copy numbers per liter of water for the CAD-A gene of each sample, and then the number of genes detected per sample. The CAD-A gene codes for cadmium resistance and is very common. The next highest correlation was between the E. coli values from the call alert and then the number of genes detected, and that had a correlation value of 0.48. You can see that transmittance has a bit more of a stronger negative correlation with a lot of these, meaning that the water that was essentially clear and had a larger transmittance, the less likely it was turbid or had a larger total coliform. So we see some of the kind of more general knowledge about what makes water have a water that has a, or what factors that are associated with a higher microbial presence in water, like higher turbidity, higher E. coli and total coliform presence. And then, but it's not strongly correlated. The next, I did a PCOA plot for all of the samples. PCOA is a method to explore and to visualize similarities or dissimilarities between data. I pulled this up and said, oh, it's a Picasso painting. It's a nice variety. You can see the St. Paul site in the turquoise, more generally toward the bottom, the Ramsey County site and the brown, generally towards the right, the Scott County site in the green, generally towards the top. But overall, and then a lot of the before, like the input samples in the circle are generally placed at the top, but overall there's nothing certain and no definite trends between all of the sites. Then I did a cat plot on all of these, looking to see if there were any particular variables, the environmental factors or the physiochemical factors that were influencing the samples either way. The cat bioplot correlates the variables with patterns to see how they influence the placement of the samples. We also ran per MANOVA tests to see which of these would be significantly different. In this case, transmittance and the number of gene detections were significantly different, whereas a lot of the other factors, as you can see, they're all clustered together and you can't even read the names. So then I went and I looked at individual sites. Burger Carver County on July 2nd, there's a huge spike in turbidity. Unfortunately, we do not have call alert data for that day. That sampling was after a large precipitation event. And then looking at the number of detections for the samples collected on that day, they're higher, they're not the highest, but they're generally on the high end for my set of data. Then I made a heat map to look at and compare all of the detections from this site over the season. The samples tended to cluster by the day that they were collected. And looking at the top brackets, you can see a separation between generally more of the right-hand chunk and the left-hand chunk. And then when I look at the chart itself, I do see a split going right through the center. For the left-hand side, which is more colorful, meaning more detections than the right-hand side, even though that it exactly doesn't match with the brackets. So if we look at the samples for July 2nd, there are more detections. However, they are not markedly different than the samples from June 5th and July 23rd. And the brackets of the heat map themselves do not single them out as well. So then I did a PCOA graph on this site. Again, no solid patterns. We have the cluster on the left here. And these samples actually were not from July 2nd, were from July 23rd and from June 5th. So on the date that the turbidity stood out and the precipitation stood out, are all clustered together with the other samples. And then on dates where turbidity and call alert levels and precipitation didn't stand out, they're actually clustered a little bit differently. So then I ran a cat plot on these. Out of these, the number of detections was the only significant factor. And you can see coliform and E. coli influencing more of the samples that were placed up, turbidity and transmittance as inverses of each other, influencing more of it downward. And then precipitation, total chlorine, and the number of detections influencing the placement on the far left. I tried the same thing with a different site. Again, for Scott County on August 22nd, we have this big spike in turbidity, and then also in the log most probable number of E. coli values. There was a precipitation event on August 20th, 2 days before. And then looking at the number of detections for these samples, they're not the lowest that we've got, but they're not higher either. When I made the heat map, you could see that the samples for August 22nd are not matched together and are kind of dispersed throughout the whole chart. Two of these samples are on the right-hand side of the chart and have less detections. One of them is more on the left-hand side of the chart and has more detections. And overall, they are not markedly distinct from the rest of the samples. And the PCOA plot didn't also not give it any solid trends. With the cat plot, we do see some of the same things that happened in the Carver County plot with total coliform and E. coli lifting upward, turbidity and transmittance downward, and then precipitation, chlorine, and detections heading to the left. In this case, there were no significantly different factors from the Permanova I ran. Transmittance was the closest with an R-squared of 0.058, but given that 0.5 is the cutoff, it doesn't make it. So for my data analysis of my samples now, there are no obvious seasonal trends in the occurrences of pathogens and antibiotic resistance genes. This definitely could be related to our sampling method. Sampling sampling stagnant water, not always when the systems are running, and then also not always after storm events, which generally spurs the conditions that are associated with pathogen and bacterial proliferation. Because transmittance had the significantly different R² value from the permanovas, and then also to an extent turbidity as the inverse of transmittance can influence pathogen and ARG occurrences, but is site and system dependent. The advanced filtration at the St. Paul site was comparable to tap water and will provide a good basis for going forward for sites that need more advanced filtration for water reuse that is more intimate than just irrigation. The St. Paul site uses the water for toilet flushing, which is why it has such an extensive treatment system. And for our 2020 data, we got funding to sample multiple times while a system is running and see how those samples compare to one another. All right. So my research team, I'd like to thank Satoshi, my advisor, Val, who was the student who did the study before me, and then the team of Dr. LaPera, Miss Anita Anderson, and Miss Nancy Rice, and the Minnesota Department of Health, generally as a collaborator. And then our funding from the Minnesota Stormwater Research Center as a part of the Water Resources Center, and the Clean Water Land and Legacy Amendment, which provided many of the funds for this study.
All right, great. Thank you so much, Tammy. At this time, if there's questions, folks can feel free to unmute themselves and ask, or you can type a question in the chat and I'll keep an eye on that.
Hi, Tammy, I have a question. So based on maybe previous work or stuff that you've read, were you surprised by the presence of the pathogens you found? Or were you expecting more? Or can you just give me a sense for like how you interpret those results?
Mm-hmm. So in the previous study our lab did, human adenovirus was actually detected. That is not something that I have generally seen in the literature. I don't think it is generally a pathogen that is, I guess, like widely tested for. So that was a bit of a surprise to me, but is, I guess, consistent with our previous findings. The E. coli was expected. It's just there's E. coli and everything. The E. coli that was detected was general E. coli. So we also we also had assays that were particularly for the kinds of E. coli that are deadly and that have killed people. But those were not detected, as well as Shigella. That I guess is a bit surprising coming from the system that has the most extensive treatment. But it's also not, it's not It's not chlorination. So it wasn't too, it wasn't shocking. And it's also not it is a bacterium that does have, or can become antibiotic resistant, but is generally easily treated with antibiotics. So if you got it, if I got it, I wouldn't be like super concerned generally. Overall, my results in the fact that I have not detected a lot of pathogens is, I think, good or promising. It's more about the kind of antibiotic resistance gene presence, and them as a antibiotic resistance gene as an emerging contaminant, and what exactly does that mean in this situation, and what is the, there's more solid like pathogens kind of like pose like an obvious risk, but like what is the risk posed by antibiotic resistance gene presence? There's just less concrete information and less like I don't really see like a consensus on it in kind of situations like this where it's it's out there in the environment.
Great, thank you.
I have a quick question. Is the E. coli that you're looking at, is it from humans, or is it just E. coli that's in the environment in general? Or did you figure out what the source was, or is it?
So Satoshi, I don't know if you know off the top of your head, but from what I understand, that this E. coli is, it does not, we did not test for, or we did not include source tracking genes on this particular study. So we don't necessarily know the origin of the E. coli, and that we just know there's E. coli.
If you had to guess, where do you think most of it's coming from? Is it animals or humans, or what? Or you don't have any idea?
My guess is given that a lot of the sampling sites or a lot of the water drew from stormwater ponds, would say that definitely animal could be a source. Potentially there's a chance of human contamination, but I think animal is more likely.
Yeah, I was wondering if septics or anything like that would be involved with any of this, but.
So most of the sites were in, I guess, like urban or suburban areas, where they're like individual houses would not have septic units. I would have to, I'm thinking of two of the sites in Scott and Carver counties, where it's, more like a bit more rural. But I'm also, unsure.
Okay.
In terms of that.
It's interesting work. A lot of data and analysis. Really was good.
Okay. Thank you.
I have a question. I think it's a follow-up to Carl's. So when I was out walking my dog this weekend, I saw a lot of like yard signs that specifically said like clean up after your dog. Like there was some sort of, I don't know if it was like a public health awareness campaign, but does that have like a major impact on stormwater? Is that kind of why would you guess that was why there was so much kind of advertisement around it?
That is definitely one of the reasons, is that it is, other than microbial contaminants, it's also like, let's say like nutrient contaminants, and that eventually the dog poo will, or at least some of it will, get into stormwater and then eventually end up in the receiving bodies of water. It's also smells and then just doesn't look good. So there are a lot of different reasons for wanting to pick up dog poo. But yes, that is also, that is why that is stressed. That is one of the reasons why it is stressed.
Any other questions for Tammy?
Thank you all for coming.
Yeah, thank you so much for speaking to us today.
Thank you. Good seminar.
Yeah, thank you for being here. Again, all the recordings will be on the website, so please check those out if you missed any, and we'll see you in the fall.
Great.
All right.
Thank-
Good.